right. So thank you everyone for joining today. For those of you who don't know me yet, I'm Alex Lemens, the director of our Molecular and Genomics Informatics Core. We also have online Pat Soteropoulos from the Genomics Center. He also, you may know, has the kind of the full suite of sequencers that can handle all the single cell workflows, such as the ones we'll be talking about today, offered by Parse. But before I just turn it over to them, just a real quick general housekeeping items. If you have any questions, please drop them into the chat or the Q&A box. I will monitor it as we go along. So the Parse team will try to answer some of the questions on the fly. Anything that's not mentioned during the talk, we will get to at the end. But with that, I'll turn it over to the Parse team to learn more about their awesome single cell workflows. So Mo, take it away.
Alex, thank you very much for allowing me to join. Everyone, my name is Mo Blackman. I'm the Senior Sales Manager here at Parse Biosciences. I am joined by my colleague, Dr. Andrea Dottie, who's our Senior Field Application Scientist. At Parse, we offer free training, free support, free bioinformatics, and our team of professionals like Dr. Andrea Dottie here are there to support you guys if you would like to talk about experimental design, assay optimization, training on the whole workflow. We're here to support, as well as bioinformatics. Let's jump into the presentation here about Parse. What I'm going to do is tell you a little bit about our technology, kind of where we came from, some differentiating factors, if you will, and tell you about an exciting new opportunity we have, which we're calling the Core Grant Program in conjunction with the Rutgers Genomics Center. So let me start by sharing my screen. Great. Well, let's start with just a little bit about Parse. For those of you who don't know, we are a single-cell RNA sequencing platform. What I mean by that is, and can you see the Parse background? Is this sharing correctly? Excellent. We are a single-cell RNA sequencing platform, and what I mean by that is what our platform does is it takes you from a single-cell suspension to sequencing-ready molecules that would go into an Illumina sequencer, for example. You can think of our platform as a direct replacement for the 10x workflow. We are essentially barcoding or indexing the transcripts in the cells. We're a relatively young company, but we've had a wonderful reception into the genomics industry at Live. We're now operating in over 350 labs around the world. We have support in Europe, parts of Asia, Australia. We're adding new labs every week. We're adding new countries every month. It's a really exciting time to be here at Parse. We're also adding some new products every quarter or so, and I'll tell you a little bit about some of the future products that we have coming out towards the end of this presentation. Where does Parse fit in the industry? Well, the single-cell genomics industry really isn't that old. If we go back even 7 years or so, people were doing hundreds of cells at a time. They were essentially trying to put one cell into one well. Other microfluidics platforms came out maybe about 5-6 years ago, kind of blew up the single-cell genomics industry, kind of set what I like to call the gold standard of 10,000 cells per sample. Really nice sample size. But where Parse comes in is scalability. We have the ability to operate at this scale. We can process and barcode 1 million cells across up to 100 samples and offer a true 1 million cell assay. Our platform consists of 2 parts, fixation and then barcoding. With fixation, you would start with a single cell suspension. You fix your cells, your samples. You put them in the -80 freezer. When you're ready to proceed forward with barcoding, you pull those samples out of the freezer and you go into one of our 3-size barcoding kits. These barcoding kits, for the most part, are the same workflow. They're the same chemistry, and for the most part, the same efficiency. The main difference between these 3 different barcoding kits is their scalability. We offer the Evercode Whole Transcriptome Mini Kit. This has a total capacity of 10,000 cells for your entire experiment, and you can split that capacity across your samples, 1 all the way up to 12 samples, in any proportion you would like. For those who are ready to scale up a little bit more, we have the Whole Transcriptome Kit. This can process and give you a true output of 100,000 barcoded cells or nuclei, and you can split that across 1 to 48 samples in any proportion you would like. So for example, you could do 10 samples with 10,000 cells each. You could do 20 samples with 5,000 cells each, or if you maxed it out with 48 samples, you'd be seeing around a little over 2,000 cells per sample. Finally, we have the Whole Transcriptome Mega Kit. This can process up to 1 million cells and up to 96 samples. Let's say you maxed that out, you did 96 samples, 1 million cells, you'd be looking at a little over 10,000 cells per sample. So this is really where Parse comes into play. We have this highly scalable platform, allowing you to collect multiple samples over multiple time points and then barcode and sequence them together in one larger, cleaner data set. This approach, as I just shared with you, is highly scalable. It's also very flexible. What I mean by that is our first step is fixation. So you fix your samples, you then store them in the -80 freezer. This allows you to collect samples from multiple time points, from multiple locations, transport them around, and save them until you're ready to move into barcoding. With other technologies, sometimes there's logistical constraints, which means you have to get your sample in, immediately barcode it. This is fine in some circumstances, but of course requires a lot of logistics. But it's also nice to just be able to have infinite flexibility. We also don't have any custom instrumentation. Our entire platform is essentially done in 96 well plates, as I'll show you in a moment. Let's see, we have little purple boxes. Sorry, I have them put away. That's our entire platform. There's no custom instrumentation. It's basically a set of reagent kits. And as I'll show you a little further in the presentation, we have unmatched sensitivity. As I mentioned earlier, we start with a single cell or a single nuclei suspension, and we bring you all the way through to sequencing ready molecules. How that works is you'd start with the same single cell or nuclei suspension that you would with other genomics platforms. You would then pass them through our kits. We have 3 different components to the kits. We have fixation, then barcoding, and finally library prep. Once you have those sequencing ready molecules, you'd send them into a sequencer. We work with Illumina, we work with PacBio and Oxford Nanopore if you're looking to do long read sequencing. And then finally, we offer a full data analysis and visualization platform. It's included for free with purchase of any of our kits. Whether you work through the Genomics Center or directly with Parse. If we take a look at the conventional single cell approach, we can say that they kind of start with a single cell suspension, as I mentioned earlier, similar to Parse Biosciences. But this conventional approach uses co-encapsulation. They use a microfluidic system to accomplish this. They co-encapsulate a cell, hopefully a single cell, with a set of barcodes inside a container. Oftentimes, this container is an oil droplet. They can then break these cells open and append these barcodes to the end of the transcript so they can understand the cell of origin of each transcript by the function of the barcode sequencing at the end of the transcript. This is conventional single-cell RNA sequencing. How Parse works is a little more elegant. We're, again, starting with that same single-cell suspension, but we're using biochemistry to accomplish the same thing. Through our fixation process, which is a 30-minute protocol, we fix and permeabilize the cells. Now, what this does is it anchors the RNA inside the cell while chemically poking holes in the membrane, which allows us to put our own DNA barcodes and enzymes through the membrane and then act on the RNA. This is really the crux of the whole technology, using the cell itself as a container for the DNA barcoding or indexing. This process, split pool combinatorial barcoding, or split-seq as it's called, was first published in Science in 2018. Alex Rosenberg, the primary author on that paper, and Charles Rocco, the secondary author on that paper, are our CEO and CTO, respectively. This approach of using the cell as a container for the DNA barcodes instead of a microfluidic system means we're not restricted by cell size. Cells that might be too large and get clogged in a microfluidic system work very well with Parse, i.e. neurons. Also cells that maybe are just too small to flow through a microfluidic system. Again, no microfluidics, not an issue for Parse. This allows us to work with a pretty wide array of cell and sample types. Here we're looking at a variety of cell lines we've worked with, some human tissues and mouse tissues, also some miscellaneous tissues, zebrafish, oocytes, C. elegans, Drosophila. By no means is this an exhaustive list, and we're adding to it every month or so. As I mentioned, the first step is fixation. So you're going to fix your samples, put them in the freezer. When you're ready to do barcoding, you'd pull them out of the freezer. And this is the part of the workflow that the Genomics core will perform for you. So what they're going to do is they're going to load your samples into this first plate. This first plate is filled with unique DNA barcodes. Each barcode is unique to each well. This first barcode, it's also used as a sample identifier during downstream computational analysis. You might be saying, hey, this is great. We've identified the samples, but we haven't actually uniquely barcoded any of the cells quite yet. Well, we call it split pool combinatorial barcoding for a reason. There's lots of pooling and splitting steps. Here in step 2, we pool all the cells, all the samples from all 96 wells together. And then in step 3, we split it to a second round barcoding plate, again, filled with unique DNA barcodes. The cells here, through this split pool process, they're starting to take a unique path. And this path is starting to be documented by the combination of barcodes at the end of the transcripts. This cell, for instance, it originally came from a blue well in the first round barcoding plate, whereas this cell over here, it originally came from a pink well in the first round barcoding plate. But now all the cells in this well are getting a yellow barcode appended to the transcript inside the cell, and that pathway is starting to uniquely be identified. We repeat this process a total of 4 times, pooling and splitting the cells. The third round plate is very similar to the second round plate, filled with a new unique set of 96 different barcodes. And of course, there's ligation steps between each of these. We then have a 4th barcode applied in these sub-libraries. And this 4th barcode is also a standard Illumina sequencing indexer, so we can be sequenced on any Illumina machine. We can also switch out this 4th barcode applied here for PacBio or Oxford Nanopore barcode. This identifies individual cells, but we also have a 10 base pair degenerative sequence between the 3rd and 4th barcode. It's not really represented here in this graphic, but that 10 base pair degenerative sequence in combination with the 4 different cell identity barcodes that we have here allows for individual unique transcript identification as well. Now, with these sub-libraries, there's a couple of really cool things that you can do with our platform that you can't do with other platforms. I want you to imagine, for instance, you're working with a new sample type. You have no idea what the sequencing efficiency is. It could be you need 20,000 reads per cell. You could need 30,000 reads per cell. You need 50,000. You don't know. With other platforms, you kind of have to take a guessing approach. You say, I don't know, let's do 45,000 reads. That sounds like a good number. Hopefully, you're not overshooting, oversaturation, and wasting those sequencing dollars, or even worse, under sequencing and missing some of your really valuable data. Well, with Parse, you can take a single cell sublibrary if you'd like. You could load as few as 100 cells into this cell sublibrary. You can run this on an iSeq or MySeq, a very affordable sequencing run. Generate a downsampling plot. In fact, a whole bioinformatics output from that small, affordable run. But really all you need is the downsampling plot and saturation curves. And you can use that to pinpoint the depths of sequencing that you need for the rest of your 15 cell sublibraries before you run your 950,000 remaining cells on a NovaSeq here on your more expensive sequencing run. This can potentially save researchers a lot of money pinpointing that depth that they need and making sure they're very efficient with those sequencing dollars. Now, altogether, after you've done this barcoding process, this is the mega kit illustrated here on the screen for you. You're going to be generating on the order of 14 million different barcoding combinations. That's 96 different barcodes from the first 3 plates, 16 different barcodes from these sub-libraries here, for a total of 14 million different barcoding combinations or different pathways that these cells can take. And with this many different barcoding combinations or pathways, that's how our technology unlocks the ability to profile 1 million cells and still have minimal doublet rates. For those of you who aren't familiar with doublets, doublets are when we see the same cell being barcoded, 2 different cells being barcoded with the same cells, and that would give us confusion as to true single cell identity, and would be considered a doublet. Here we're looking at a standard species mixing experiment. Ideally, what you want to see is each one of these cells is either falling on the x-axis or the y-axis. That would indicate that we have reads coming from one cell, but not from one species, but not from the other for each particular cell. However, if you see something like this gray dot in the middle, this means that we're detecting transcripts from 2 different cells, which we know is not biologically possible, and therefore what we're really looking at is a doublet. So what are Parse's doublets? Well, a lot of people say, what's the big deal with doublets? I can remove them computationally, and that is true. Better to have a cleaner data set to start with. But the real problem with doublets is that you're paying for those sequencing reads. And because they're doublets, you're double paying for them just to throw out those reads. With Parse, let's take a look at our medium-sized kit. Let's say you max out the medium kit, the 100,000 cell kit. Here, we maxed it out. We ran 100,000 cells through. You'll notice that we're getting a collision rate here in the center of about 1.7%. This doesn't account for mouse-to-mouse or human-to-human collisions. So the true doublet rate in this 100,000 cell experiment is 3.4%. If you did less than 100,000 cells, you'd get a far lower doublet rate as well. Our mini and our mega kits have a different amount of pathways and capacity, and they have collision rates of 2.3 and 3.2% respectively, which gives them a true doublet rate when maxed out of 4.6 and 6.4%. We find this to be a third, sometimes even a quarter, as few doublets as other droplet-based methods. Again, allowing you to not throw out those doublet, those valuable, expensive sequencing reads. When you do run a single cell experiment, it's going to be critical for you to understand the proportions of the cell populations that you put into the original experiment. And with Parse, what we looked at, we took PBMCs from a healthy donor. We got these from an upstream supplier. They ran flow at the protein level. Then they sent the samples to us, and we ran our entire single cell RNA sequencing experiment on all those same samples. You can see here we have very high correlation between our own internal data and the data reported by our upstream supplier. And this assures that we don't have any kind of bias to any specific cell type while running our single cell RNA-seq experiment. This is a different case example here. This one, again, using PBMCs. Here we were looking at 24 different donors, 40,000 cells per sample for just over 1 million cells. Now, I like to think of this as our brute force approach. Let's say for whatever reason you can't do some type of sorting or enrichment or optimization to enrich for a specific cell population through your given experiment. With Parse, for most experiments we can certainly do that, but sometimes with logistical or scientific limitations. Like I said, I think this is our brute force approach. Say you can't enrich. You can still fairly economically run 1 million cells through our platform. And then even if that cell is 1% of your target population, you're still going to be seeing 10,000 cells from that target cell. So if you're looking for rarely occurring cells, basophils, plasmablasts, things of that nature, we can potentially obtain them through this brute force method, running 1 million cells through. This, again, a different case example. This was a paper that the Allen Institute published. Here they were looking at stem cells differentiate into cardiomyocytes. They had multiple time points for them across 90 days. They were looking at multiple cell lines and different differentiation protocols and replicates. So in short, there were many samples collected here at each one of these time points. Now, normally this would be 6 to 7 separate 8-lane experiments and other commercially available instruments. But with Parse, they took 48 of their 55 samples that they published in their paper, and this is linked on our website. And they ran 48 of those initial samples on one WT kit. I believe they ran the others on another WT kit. And what they were able to do was pool all these samples together, barcode them during one run, sequence them during one run. This, of course, saved them time. It saved them some money, but also reduced their batch effects because they were pooling all of these samples together. It's a relatively simple experiment, but really emphasizes the versatility and scalability of Parse's platform, the split pool combinatorial barcoding approach. As I mentioned, we originally published this approach in Science in 2018. At that time, we were seeing around 15,000 unique molecules per cell. We're pretty happy with that number. There's certainly room for improvements. Since then, we have optimized the platform. We've added a whole support suite. People like my colleague, Dr. Andrea Dottie here, as well as our bioinformatics team, and a whole slew of other support systems for you. We've also optimized the chemistry. We're now seeing around 90,000 unique molecules per cell. Now, full disclosure, this is HEK cells. They have a pretty high RNA content, and this was sequenced to saturation. So you would hope to see a good amount of transcripts detected. So a lot of people say, great, 90,000, I'm really happy with that number, but what's the sequencing efficiency? That's the other half of the picture. When we start to talk about chemistry, in this next slide, we'll get to that core question of how efficient is Parse's libraries or sub-libraries once you do sequence them. Here I'm showing you 4 different downsampling plots, 4 different experiments. The x-axis is the number of raw Illumina sequencing reads used per cell, and the y-axis the number of total genes detected per cell. PBMCs and 3T3 mouse cell line, these were run using whole cells. Mouse brain and mouse heart was run using nuclei. The gray line is representative of 10x's most recent chemistry. Across the board here, Parse Biosciences is outperforming 10x's most recent chemistry. And to really illustrate this point, what I like to do is kind of draw the semi-straight line across here and say, okay, you're running PBMCs in the 10x platform, and you want to get, we'll round up to 2,000 genes per cell. You're going to need about 40,000 reads per cell. With the Parse platform, you can accomplish and capture that same level of information with about half as many reads. This sequencing efficiency, a lot of people say, what's that account? How do you account for this? Where do you get there? And of course, it's a different approach, it's different chemistry and so forth. But one of the main reasons, the main differences here is we are using both poly-DT and random hexamer primers during our RT step, which is in the round 1 barcoding plate. This allows us to get some 3' information. We tile along the transcript. We get some 5' information as well. We're getting whole transcriptome coverage. We're burning the candle from both ends, and that is why we kind of get a 2-to-1 increase in genes and transcripts captured over competing technologies. This sequencing efficiency, coupled with the fact that we have 1/3 to 1/4 as many doublets, and that you can pinpoint the depth of sequencing you need before you sequence all of your cell sublibraries, all these 3 things together potentially let you save a lot of money on the back end with regards to sequencing. And for any of your single cell projects, generally, historically, sequencing is about half of the budget. So very nice to be efficient there. As I mentioned, we use standard Illumina sequencing indexers in that 4th and final sublibrary there, the 4th and final barcode applied in our sublibraries. We can be sequenced on any Illumina machine. If you're running, say, just 1,000 cells, a MySeq might be enough for you. Maybe you're just doing one of the cell sublibraries in an initial run. If you're scaling up to 20,000, 50,000, 100,000, or a million cells, you're going to need, of course, a higher level sequencer. We do recommend that you use sequence one of the smaller cell sublibraries first to kind of pinpoint that depth. But let's say you say, hey, I don't even have my samples yet. I'm applying for a grant. You need to have a budgeting. We recommend around 20,000 reads per cell, just generally, if you don't know, if you don't even tell me what your sample type is. Now, across various sample types, HEK cells, 3T3 mouse cell line, nuclei samples, at 20,000 reads per cell, by no means saturation, we're still seeing a great amount of transcripts and genes detected. Couple of other really cool things with the Parse platform. It really comes down to flexibility. These fixed samples, let's say you have plenty of starting material, you can fix these samples. You could take an aliquot, you could run it on our mini kit. Take a look at the data, apply for a grant, make sure everything's really good. Then 6 months later, you can take a 2nd or a 3rd aliquot out of your freezer and run it on our medium kit or our mega kit or both. This allows you a lot of flexibility and scalability, ability to start small and scale up later. It also provides you a lot of insurance because you have a portion, an aliquot of that fixed sample waiting in your -80 freezer for you. Our fixation kits, they come in 2 different flavors, whole cells or nuclei. The same fixation kits are paired with all 3 size barcoding kits. Once we fix those samples, we don't really care. They're treated the same way after that. Now, while working with the core, again, lots of flexibility, most researchers would elect to do their own fixation. This means we would ship the fixation kits directly to your lab. You'd collect your samples over multiple time points. You fix them, you put them in your -80 freezer. Now you've collected 10, 20, 100 samples. You pull them out of the freezer, you put them on dry ice, you walk them over to the Rutgers Genomics Center. They purchase the barcoding kits directly from Parse and perform the Parse barcoding and library prep workflow. And if you'd like them to, the sequencing as well. This gives you lots of flexibility and logistics for just grabbing those samples, storing those samples, collecting them over multiple time points, maybe working with a clinical partner. Lots of flexibility there as well. Now, a lot of people say, okay, great. I've put 96 samples in my freezer. I collected them over 6 months. I'm about to pull them out. They've been in there for different amounts of time. I'm a little concerned with data quality. How did the samples deteriorate over time? And that's certainly a fair question. Here, we looked at fresh samples, samples that were fixed and then immediately barcoded versus ones that were fixed, stored for 6 months, and then barcoded and then sequenced. For both median transcripts per cell and genes per cell, we're detecting just over 97% of the same amount of information. Again, I'd point out, this was 2 separate barcoding runs and 2 separate sequencing runs as well. So not only do these 2 charts speak to the minimal loss of data quality after storing our samples for 6 months, but they also speak to the high reproducibility of the platform at large. After you do fixation, bring those fixed samples over to the core here, they will perform barcoding for you. Again, the barcoding kits come in 3 different sizes. The mini kit, total capacity of up to 10,000 cells. You can split that full capacity across your samples in any proportion you would like. Let's talk about that for a moment. If you maxed out our mini kit to 10,000 cells, 12 samples, you'd be looking at only 800 cells per sample. Relatively small amount. This is better for proof of concept studies if you're really maxing it out with 12 samples. If you did just one sample, you could designate the full capacity of 10,000 cells to that one sample. Or have 2 samples, 3 samples, all the way up to 12. If you're working with our medium-sized kit, the Evercode whole transcriptome kit, total capacity of up to 100,000 cells. Again, if you split that across 48 samples, you'd be looking at about a little over 2,000 cells per sample. If you told me, hey, I need 10,000 cells per sample, well, then you can think of this whole transcriptome kit as a 10-sample kit. 10 times 10,000 gets us to 100,000. Finally, for those people really looking to scale up, pull out all those samples from your bioarchived system, we can profile 96 samples at a time across 1 million cells, giving you over 10,000 cells per sample. And this is very economical as well. There's multiple stopping points built into the protocol. No more 13-hour days for your core. So they're going to be a lot happier and easier to work with. There's logistics become a lot easier to work out. We also have multiple QC checks through the entire protocol, just to make sure that before you go on to the next step, you successfully completed the first one. Finally, we have the library prep kit. This converts those sub-libraries that we've been talking about to sequencing-ready molecules that would go into an Illumina sequencer. Once you do sequence your libraries, you're going to get FASTQ files back from your sequencer, just like you would with any other sequencing run. You're going to pass those FASTQ files through the Parse pipeline. Our pipeline is free. It's as is pipeline training and pipeline support. We'll go as far as running the pipeline for you if you have any trouble doing it. The pipeline can be downloaded. It can be locally run on your cluster. It can also be run on the AWS system and very soon cloud-based computing platforms as well. There's 2 outputs to the Parse pipeline. The first is a cell by gene count matrix. This serves as a great input to downstream analysis tools. ScanPy, Syrah, we play nice with both of them. This is also really nice if you've already done previous runs on other platforms. You can combine new Parse data with your historic data in Syrah, and that way you're not losing anything. The second output is the Parse experimental summary report. A few things here. It's HTML-based, makes it very easy for you to share with your colleagues, both internally and externally. It's fairly user-friendly. We show you a few things here. We show you some relevant sequencing stats. We show you some downsampling plots and some saturation curves. We also show you some heat maps, how many cells went into each well, how many barcodes went into each cell. We report out preliminary clustering, which you can see here over on our cluster map. We also have this differential expression table, which links to the NIH gene database. On our cluster map, you can export these images. They're high resolution, great for publishing on posters. You can type in your target gene. You can see where it lands on that cluster map below. I believe here we're looking at an excitatory neural marker. This is our customer support suite. This is a gated section of our website. Right now, if you went on here, you would see some information, but only about 10% of it. The really good stuff, you need a password for. And to get that, you just need to purchase any of our kits, and we give you full access to the entire support suite. This includes pipeline documentation, downloading the pipeline, videos, tips on the whole wet lab workflow, all of our protocols. And it's also a place that you can interact with our support team, whether that be our field application scientists or our bioinformatics support team. What else is on the horizon? Well, we talked about our current kit, the Evercode whole transcriptome kit. Very proud of that kit, but there's certainly some additional technologies that we want to explore. And we've announced that we'll be very soon releasing, hopefully before the end of the summer, a focal barcoding kit, a attack-seq kit, a gene panel kit that will be enriched for a set of targeted gene panels. We have a standard panel, and we also have room on the panel to customize it. And we have an immune profiling kit coming out as well. The gene panel enrichment kit and the focal barcoding kit, what these basically will give you is a second set of sub-libraries in addition to your standard set that comes with a regular kit that are either enriched for a targeted gene panel or a specific transcript. Lastly, I would like to tell you about a really exciting opportunity that you have to work with Parse Biosciences. There's 2 things. The first is for for anyone that's on this call today, thank you very much for attending, for giving us your time. We wanted to say thank you. And so I'd like to offer you a free cell fixation kit with the purchase of any of our other kits. And if you'd like to take advantage of that, please just through the chat feature here in Zoom, send me your email address and I'll send you a voucher email for a free cell fixation kit or nuclei fixation kit with the purchase of any of our barcoding kits. And that'll be good for 6 months and you can use that whenever you're ready to proceed. I also wanted to tell you about the core grant program, we're calling it. Now, what the core grant program is essentially an opportunity for you to try out the entire Parse workflow without having to pay for any reagents or any labor costs. We are inviting you to submit a abstract. This is 300 words. It can be a little bit more. We didn't actually put a limit on it. We did write 300 words in here, but please feel free to email me with supplemental information or anything along that nature. You would tell us a little bit about a project of your choice. The deadline, it closes let's see, 2 weeks from this Friday. So I think that gives you about 16 days, Friday, July 8th. So please log onto our website, submit an abstract. What we're gonna award to one of the abstract winners, we'll read them as a team, vote on them, and then award a winner. Award a winner. And we will give them a free WTK kit. This is good for barcoding 100,000 cells or nuclei. We'll either give 2 cell fixation kits or 2 nuclei fixation kits, and this will be good for up to 8 samples. We also provide full data analysis and support and training on the Parse fixation protocol. The core has generously offered to perform the entire Parse workflow, barcoding and library prep at no charge, so no labor costs there. However, the winner, the individual recipient, will be responsible for purchasing an S4 flow cell. So there is some cost should you win, so please take that into consideration when you fill out this landing page. If you would like access to the landing page, please, again, within the chat feature here, just send me, you can either send it direct or through the whole group, send me your email and I will send you a link to fill out this abstract and potentially be able to be a recipient for this core grant program. Lastly, I wanted to turn the microphone really over to your team, to everyone on this call here, to see if there's any questions that I can answer or that Dr. Andrea Dottie can answer, our senior field application scientist on the call. Alex, yes. Do we have any questions?
We had a couple questions so far in the chat that Andrea's already answered.
Yeah.
I guess if you want to just go through them real quick, just that way everyone can hear the answers, because I know some of them were,
Sure.
I was writing down questions the whole time myself, and some of these were actually paired very nicely with the questions I was planning to
No
ask.
problem. So we had a couple questions about working with delicate cells, for example, like immune cells, like neutrophils, that actually have the lovely ability that if you process them too many times, right, they happen to activate and degranulate and form lovely sticky nets in solution. And so you guys know my background is that I'm a PhD in immunology. I was a faculty member at the University of Florida for a while before I joined the Parse team, and I ran the flow core. So I have a lot of experience in working with a tremendous amount of sample prep, and I ran a whole TEDx pipeline that was in my lab, and I also had a beauty Rhapsody in my lab. So I've worked with a lot of these weird activating kind of cell types, and I know how unhappy they are when you process them through GEMS. So yes, our fixation protocol actually works very well for neutrophils. So that's the first thing. Again, it is all about the front end handling, right? Being more delicate with them so we don't activate them, and moving fairly quickly from the time that you actually get them out of the, blood fraction if you're doing PBMC preps, or if you're growing them in culture, being gentle, getting them back up out of the culture system and into the fixation media. But as long as you process them delicately, there's no issues with them as far as them kind of becoming a mess later on. The other question that we had from one of the people who was attending was about site-seq. So again, we don't have any internal data right now that says that we can support 100% site-seq, but we do have a lot of customers trying this in the field. We're kind of waiting on getting that data back a little bit. So my answer is a little one of those that I think we should have no issues with that. But I would recommend that you test it, just because, again, it's not something we currently have a bunch of data on to support to say absolutely yes or no that that works. But we have some customers who are in process of doing that. And as far as I know, tentatively, preliminarily, we don't actually have the data, but we have heard that this has been successful. So I think that as long as you're willing to do a little bit of optimization with the fixation on the front end of that, would be my recommendation if you're doing something like that. And then the third question had to do with hashtagging, because you're pooling the samples. So the beauty of Parse Assay, I think, and having been a person who's done 10x and hashtagging, is the nice part is that you actually mix all your samples together, right? So in the first plate of the barcoding process, you're actually indexing all your cells and all your samples with a unique molecular index for the sample. So you don't actually have to hashtag, because you're doing the first part of that. Indexing happens where you're going to annotate like a UMI as a sample index. So when you go back and look at the data analysis, you can parse those samples out without having to use hashtagging. So it's actually a really nice feature. And when you mix all the libraries together and you sequence them, you're getting a pooled mix of all of your samples together. So when you look at them computationally, you can see those individual barcodes that were barcoding your samples. And because you use all the wells in the first plate, you could actually split a sample across 3 or 4 wells and have 4 technical replicates of the same sample with different UMIs that you can reconstruct on the back end. So it's a really nice feature that I like a lot, having to kind of bypass that hashtagging and worrying about that. So those were the questions that I had. If anybody else has any, I'm more than happy to answer them.
Okay. Sure. I got a quick technical bioinformatics question. Just you mentioned how it's very compatible going into kind of the downstream analysis tools like Seurat and ScanPy. So I'm guessing your kind of your parse initial pipeline is equivalent to 10x's Cell Ranger. So then the output files, are they, when you describe the matrix, are they more of a compressed format like an H5 format, or are they your classic TSV CSV type of format?
CSVs. So you're just going to take those and then bring them into whatever else other kind of secondary pipeline you want to use. So we have people using different informatics things from GitHub, and they tend to work. You just have to do the processing on the front end through the parse pipeline. And one thing that's different about our informatics pipeline is that you don't concatenate the files. You run each library as its own file first, and then you combine. We have a combine mode at the second half of the pipeline that combines the samples together and concatenates them at the end. So that is a distinct difference than what you would normally do in 10x.
So by concatenating them, are you kind of just merging the data together, or is there some sort of integration algorithm as well that kind of can take into account that both? Okay.
Both. It does both. Yep. And again, I'm not a computational biologist, so I'm going on what my FAS, who is on our team. We have an informatics FAS who actually works with this pipeline. And if you guys work with Parse, we do 2 parts of the training. One, you get me for all the front end of the technical part, all the way up through library construction, and I'm your troubleshooting kind of guru for all problems that happen in the assay. But once it comes to the data analysis, we have a combined call with our informatics FAS. And then he handles all your back-end computational biology kind of questions, because I know what my strong suit is, and it is not that. I'm good at it to a point with Cell Ranger, but I'm also not the expert. I don't write code. I'm pretty honest.
Alex, perhaps you could share a little bit about the bioinformatics services that you offer through Magic.
Yeah, I mean, they're kind of just right now the standard single cell offerings. Usually we go from, depending upon what you need for your studies, because I know a lot of people have either bioinformaticians on staff or, kind of their own collaborators. So a lot of times we handle the upfront primary analysis, doing that initial deconvolution, in this case, the barcodes into that matrix file. So that way you don't have to worry about having the much heavier compute that I imagine is required for that. Because fortunately we do have, full access to the Emerald system and a couple of dedicated nodes to process all of that data. But from there, we also do have collaborative services and regular fee for services for doing kind of the secondary analysis, where if you need to take that into Seurat or ScanPy and starting to actually dive into your data a little bit more, because I haven't played with the Parse system yet, but I know that's one of the limitations for the 10x system is their cell browser has not been adequate for the needs that we have seen. So we'll have to play with the Parse one as well once we get some data in house. But a lot of that ends up going into kind of our workflows for either Seurat or ScanPy, and then doing a lot of times, since it's transcript based, we have a lot of the like wild type versus control, treatment versus control, knockout versus conditions, things like that, which really usually requires a lot of kind of downstream workflows. And then a lot of times we end up bringing that into more of a kind of quasi pseudo bulk analysis, which fits your kind of your traditional canonical RNA-seq workflows. But yeah, all of that is available. So if anybody wants to talk about that, we'd be happy to. But I'm excited to see some of this data come in hand, because I'm on the data side, so I like just playing with it.
Well, and I mean, I think having that resource as part of the core is really kind of critical, because I think that's a really good partnership, is that, with your own independent research, bringing it to a core facility. I was at UF. We also had that. We had a next gen gene expression core and a bioinformatics core. So I worked very closely with them, and those partnerships are so important, because at the end of the day, everybody has their strong suit, right? And I think it's really important to have all of the people who are doing the biology be part of those conversations, but not all of that is our strong suit, right? And having experts we can go to, and if you have that inside your own resource, that is an incredibly useful tool for your people to have on hand.
Yeah, 100%. And it's also good, kind of on both sides of it, at least knowing what is in the other sides. Just because I know there's times where I'll look at the data and say, hey, something looks a little funky here. Having an understanding, hearing from Mo how this entire process works, knowing that, okay, maybe there's an issue in this step of the process. Right. And kind of on the flip side, Pat and her team, which is great to work with them, and they are fantastic to work with. They always know now, okay, well, when you're designing this experiment, you have to have these considerations in mind to ensure that you have statistically, validatable results on the back end, which that hand in hand needs to happen.
Yes.
We do have one new question regarding sequencing configurations and requirements. I'm going to turn this back over to Andrea to answer. Maybe you can discuss a little bit how we would set up sequencing and which flow cell and so forth.
So we are this is our read structure for the Parse kind of construction wise. So we have read 1 would be your mRNA transcripts, or read 2 are where the barcodes and the ligation kind of linkers are. And so we are a single index library. Now, these can be combined regularly with dual index libraries, as you would with pooling assays for big runs on a flow cell, with no problem. But we do want to make sure that people do understand that we are a single index right now. The other thing that makes us a little unique, other than what you would normally do for a 10x pipeline, is that our read 2 requires 86 base pairs instead of the typical split of 75 and 75. Now, pairing anything more than 86 to that doesn't give you any useful data, so we tell everybody to port it back to read 1. But you do have to get at least 86 programmed on the Illuminas to be able to get the correct kind of configuration information, so that you're going to end up, when you package that out and you look at all the data, you're going to reconfigure these in the pipeline on the back end and then pair them back together. But that's the substructure of what the library and the read length looks like. So we use a typical Illumina i7 index for that. Does that answer your question? I'm assuming.
I think it did, and I guess I would add that we use a 150 paired end flow cell or greater.
Yeah.
To accomplish this. I mentioned that because some people add these up, and I think it comes out to 168. The 150 flow cell actually is 168. They have a little extra capacity in there, which we take advantage of.
And typically, I think now most of the NovaSeqs are running at 200 kits or bigger. Right. Yeah. So I think that's kind of only if you're using a NextSeq or a HiSeq in there, if you have an older sequencer.
And you also mentioned, kind of just pairing off that question, how you've integrated with the long reads platforms like PacBio and Nanopore. I'm guessing that's to get a lot more kind of information on the splice forms that you might be identifying in there. Could you just, give a little bit more detail on that? Because I know we've had a few people with interest in that in the past.
Sure. So you would end up, instead of doing the last index where you index for the I7, for a standard Illumina library, you would basically insert whatever the index kit would be for PacBio or Oxford Nanopore at that section. So there's a stopping point in the protocol where you're going to do that I7 index barcoding reaction, and it's after plate 3. So you do all of our barcodes, and then the last barcode is kind of a separate kind of sub-reaction that we actually include in the I7 index, but you can substitute that for PacBio's, and our chemistry plays well with theirs and Oxford Nanopore with no problems.
Okay. And have you guys seen any data with that? Like I know PacBio, I imagine you probably have to use their hi-fi reads, and then for the Nanopore, I know they're improving their accuracy, but has that had any issues with the barcode strategy?
I do know we have that in process. I haven't, honestly, for my perspective, I actually just haven't bothered to kind of dig that out, because I'm honestly, I'm kind of buried with other training calls and client things. But I do know that we do have some internal data on that, and I can get back to you on that. So what I'll have Mo do at the end of this is connect me with you guys, and then I'm happy to send you the information that we have on that, and hopefully that will answer your question.
Awesome. Yeah, thank you.
Yeah, I'm sorry about that. Like, I don't have everything in my back pocket. I try to be pretty flexible about those things, but some things I'm like, maybe I don't have that.
I'd also like to share this. This is a paper by Dr. Liz Raboa. She has this discusses using the PacBio system and Parse together for long read sequencing, basically switching out that 4th barcode for the PacBio indexer. She also gave a really nice webinar presentation on this paper. I know I have the archive up, but she has at this point published it. She gave a great webinar about a month ago. I'm happy to share it with anyone that would like to watch it. And at the end, she mentioned while the PacBio data was great and published on it here, the Oxford Nano data and Oxford Nanopore data looked even better. So if anyone's interested in seeing this paper, I'm happy to send you a link to it. Please, again, just share your email address in the chat, because the core here is actually not sharing your email addresses, because we didn't obtain permission to do that. So if you do want to communicate with us, please let us know. Also, you can email me at a later time. My name is Mo Blackman, and my email is mo@parsebiosciences.com or Andrea@parsebiosciences.com.
And it's Andrea with an I, not an E, by the way. So.
And you can also always reach them through us, because we're in close contact with Mo and Andrea as well. So you could reach out to me or to Alex, and we can put you in touch with them.
Yep. And we will be sending a kind of a follow-up to everyone, just to kind of send the link to the recording. We will include everyone's contact information so that we can reach out and continue the conversation as you wish.
If anyone would like any quotes other than those that have already requested them, please let me know. Happy to quote any of our size kits, including the fixation kits. To give you the real short sales pitch, if you will, on it, is our platform becomes dramatically more affordable as you scale up. From the cost per cell of the mini kit, you're looking at, with academic pricing, about 43 cents per cell, which is about on par for what the industry charges. But as you scale up to the medium kit, you're looking at about 9 cents a cell, or the mega kit, only 1.5 cents per cell. This lets you do 1 million cells for under $18,000, and that's all in everything you would need from Parse. This comes out to about $187 a sample for 10,000 plus cells per sample. We find this to be an extremely economical approach, especially on the higher scale end of the platform.
Yeah, definitely. I mean, compared to some of the other library prep kits that are out there for single cell, looking at $187 per sample versus let's say 2,000-ish, it's a price savings. But you do have to also calculate in sequencing costs in addition to that.
You certainly do, as you would with other platforms as well. As a reminder, with the sequencing, we tend to allow researchers to have really efficient sequencing for those 3 reasons. 1, we can pinpoint the depth you need before you sequence everything. 2, we have very low doublet rates, so you're not throwing out those reads. And then finally, very efficient libraries once they are sequenced. I think that is all of the questions. Anyone else want to jump in with any last minute questions? I wanted to take the opportunity one more time to thank both Pat and Alex for inviting us to speak with you guys. We appreciate the time, the interest. If anyone would like to set up a private meeting, please reach out to us at your leisure.
Okay, and thank you, Mo and Andrea, for presenting today. This is very informative, and it was a great seminar. So yeah, like I mentioned before, we'll pass the recording out over the next day or two. And I'd encourage everyone to apply for the, send in an abstract to see if you can win the the free kit. And we'd love to see some of this data in-house and test it out and see how it looks, with some samples that we can work with. But thank you, everybody. And I guess with that, have a great day.
You too. Thanks for hosting us.
Bye, everybody.
Thank you, everyone.
Bye.
Bye.
Bye.